Hello everyone, and welcome to lecture 25 on sensory physiology. So in this lecture I'll be focusing on the nervous system's pathways for sensory information and primarily focusing on pain. Right, so I want to begin by talking about the brain. So we have lobes in the cerebral cortex, right, which are labeled as such. You can see here there's the frontal lobe, parietal lobe, occipital lobe, temporal lobe. And so they're designated regions of the cerebral cortex that carry out certain functions. Okay, so it's actually, the brain and spinal cord are very well organized. Now, for example, like the frontal lobe, that can carry out sort of motor processes, right? So efferent outputs to our body to help carry out motor commands, as well as higher executive functions. Then we have in the blue shaded area there, the parietal lobe, which is really the sensory portion of our brain. And then there's the occipital lobe for vision, temporal lobe for things like hearing and memory. And then down here, this isn't a lobe, but this is the cerebellum for balance and coordination. And then you can see the brainstem and spinal cord down there. And so for sensory purposes, really I'm going to be focusing a bit on the parietal lobe. All right, so when it comes to the brain, the parietal lobe is the area of interest for what we call somatosensory inputs. So somatosensory is coming from the body. So things like touch, proprioception, vibration, pain, temperature, these are all different sensation modalities that get processed and interpreted by the brain in regions of the parietal lobe. Then we have a category of special senses, and those special senses are going to different regions of the brain. So for example, things like smell, so that would be going into sort of the frontal lobe, and this is what we call through the olfactory nerves. Then you have the occipital lobe for vision, temporal lobe for hearing. Those are all sort of special senses. But when it comes to, again, the sensations, the somatosensory sensations, that's going to come into an area called the primary sensory cortex. All right. And so you notice that that's only one region of the parietal lobe, because the parietal lobe has other regions that are designated to helping sort of interpret and process that information. So that primary sensory area in the parietal lobe, if we don't have it sort of color-coded, we see the brain that looks like this, and it has all these sort of bumps and grooves in it, which we refer to as these gyri and sulci. So the gyri are the bumps or the hills, and the valleys are the sulci. If it's a very prominent valley, we'll usually call it a fissure, for example. Now, the brain has something called the central sulcus, and the central sulcus actually separates the frontal lobe from the parietal lobe. So remember the frontal lobe, all right, and the parietal lobe, all right, they'll be separated by this central sulcus, which I'm outlining here in red for you. And so that goes across, horizontally across the brain there. So you'd see it on the other side as well. And so what you have here is just after that central sulcus is what's circled for you in red here. And that's referred to as the post-central gyrus. All right, that's like this hill. And its function is the primary somatosensory cortex. It's going to pick up that information from the body and transmit it to this region of the brain that's just behind that central sulcus. Now, there's a gentleman known as Brodmann who actually looked at the cytoarchitecture, or the different cell types within the cortex itself, at different regions of the cortex, and based on their cytoarchitecture, he gave them different numbered designations. And it just so turns out that those same numbered designations, when you're looking at the cytoarchitecture, also relates to the function that it carries out. So oftentimes you'll see in textbooks and whatnot that they'll refer to certain regions of the brain by their Brodmann area, which not only is labeling its type of neuron cell type that's there, but also the functionality. So for example, in the auditory area here, you have Brodmann area 41 and 42. So there's certain types of cells there, and these are specialized cells for processing auditory information. Over here, 17, 16, that's our primary visual areas in the occipital lobe. So you can see 16 and 17 right there. All right. Now these other Brodmann areas are also used for processing information and so on, but these would be ones I pointed out are kind of the primary areas that's where the sensory information is initially coming to, and then to be processed. Okay. And so these areas, so basically if I'm to feel like sensation in my fingertip, that sensation has to go, it's an afferent nerve that's carrying that, right? And that's going to carry that information to my brain. It's going to carry it initially to this post-central gyrus area. And then from there, I can interpret that information. And so I put down there that, for example, touch is a type of sensation that's carried in what we call the dorsal column. So I'll review those pathways with you in a little bit. And then pain, spinothalamic pathway. It's a different pathway that it takes to get to the brain for to be interpreted. So we'll talk about those different pathways. Now, our nervous system, has to be able to sort of differentiate and localize that sensation. So these are two important properties. So in other words, by differentiating it, meaning differentiating it from, say, light touch versus a deeper pressure or deeper touch, vibration, or separating it from different variations in temperature, pain, something we call proprioception. Proprioception is our awareness of our body in space. And so in order to be able to pick up on all these different external sort of modalities or environmental cues, they would have to have specialized receptors for that. And so we're not going to get into all the different types of sensory receptors that are there, but there's a host of different ones. And so, for example, you'll have these receptors that can pick up on mechanical stimulation. We call them mechanoreceptors. So, for example, if you're doing pressure and touch, that can cause mechanical distension and opening up of channels that allows ions to flow, for example, and creates a depolarization in that sensory neuron. So what we're trying to do is go from a mechanical stimulus, like touch, and turn it into an electrical signal. And so opening up of channels from mechanical stimulus allows ions to flow, causes depolarization, and then an action potential. So that's like something over here. You see the fingertip. We transduce that mechanical pressure into an electrical signal. Its cell body is located in that dorsal root ganglion, so you can see that sensory information. It's the afferent neuron carrying that back to the spinal cord and ultimately to the brain. Right? It's going to take certain pathways, and we'll talk about those pathways later. So mechanoreceptors are one type. Photoreceptors for light, like for example in the retina. Chemoreceptors. So for example, taste. Or eating something salty with sodium chloride, so it's able to pick up on that and transduce a signal. We also saw chemoreceptors in the carotids and the aorta, picking up on changes in our proton levels, CO2 levels, and so on. I'll skip nociceptors for a second, but thermoreceptors. Thermoreceptors are picking up on changes in temperature. The nociceptors, so I'm singling those out right now because nociceptors are pain, so they pick up on painful stimuli. And nociceptors, they're really, I really should pull it out here because they're really a combination of mechanoreceptors, chemoreceptors, and thermoreceptors. So they're really all three types. In other words, really deep pressure can cause pain. So that's a mechanoreceptor. Chemoreceptors, as well as some increases in acidity and stuff that can hurt the stomach lining, that can cause pain. So protons, for example. Thermoreceptors, so extremes of temperature, severe hot or cold, can initiate pain. So there's special pain neurons. We refer to their receptors as nociceptors, but they're the ones that get signaled by these sort of extremes of pressure and temperature and so on. Now, before I move on any further, another interesting thing is that we also have to localize, right? So that's how the receptors help us differentiate one type of pain from another, or one type of sensation, I should say, from another. But then we want to be able to localize that, right? So if the pain is coming from our finger, we know it's in our finger or our knee and so on. And so there's varying degrees with which our neurons can sort of localize that, varying degrees of precision. Some are far more precise than others. So for example, even within pain, we're going to talk about that. Some types of pain are easy to localize. We can be pretty precise. Other types of pain are very diffuse. All right. So localizing it. Now, in terms of localization, our brain kind of already has sort of this architecture or this map of our body in mind, sort of blueprint of where all the neurons are. And so when we, for example, prick our finger, like in our right hand, when that signals, it goes to the left side of our brain, and we know that that's coming from our right finger. And so we know that partly because then the brain is already built in sort of this map of where these neurons, where these signals are coming from. So we're able to sort of translate that information to the appropriate place. So same thing if it was, pricking the toe or the knee. So it's kind of mapped out where those neurons are. And so we're able to sort of translate that information to that region of our body. Now, in that same thought, for example, if I take this guy here and his finger, if you were to pinprick the finger, yeah, you'd feel the sensation of the finger in the appropriate place under normal circumstances. However, if I were to stimulate anywhere along the neuron here, Like I say, I took electrode and just activated the neuron somewhere else along the path, not exactly at the fingertip. Where would I feel that sensation? Now I'm not talking about touching the skin and other places. Anywhere I touch the body, the brain can pick up that the sensations coming from that part of the body. So we know that. But what I'm talking about is just sort of isolating that one nerve and stimulating it. And where would I feel the sensation? Regardless of where I stimulate that neuron, okay, where along that path, it'll always be perceived as coming from the fingertip, because that's where the specialized receptors come from, and that's where the brain is sort of mapped out. When I get signals from this neuron, it's coming from that fingertip. And so that's how it's perceived. So again, if for some reason the neuron over here was to fire somewhere else along its path, it would still be perceived as in the fingertip. The reason I make this point is because it's, clinically, when we have, let's say, somebody who's like an amputee and they have this phantom limb. Right? You may have heard of phantom limb, where if somebody, let's say somebody had their right hand amputated, they may still have sensations in their fingertips. Now, it's weird because they don't have fingertips in the right hand. There's no hand there. However, even though you remove the hand, you still have remnants of the neuron that was part of the neuron that went to their fingertips. And so what happens is, by removing the hand, if for some reason we're stimulating or irritating the neuron that used to be connected to the fingertip there, like I said before, if you stimulate anywhere along the path there, it's perceived as coming from the fingertip. That's how the brain has mapped it. So in this case, phantom limb, people perceive to have pain or some other types of sensation in the limb that's not there anymore, because the neuron, the remnants of that neuron that are still being stimulated or irritated in some way, are telling the brain that it's coming from the limb that's not there. Okay, because that's how the brain perceives it. Which ends up leading to very fascinating studies about how it's going to handle that. Same type of thing if you've ever hit your funny bone. So again, the funny bone, really what you're talking about is hitting the nerve that traverses the medial portion of the arm there. All right, so if you hit that, what you're doing is you're irritating and sort of activating not only neurons that are located in that region, so you feel it in your elbow, and it's coming from your elbow, but also you're stimulating any nerves that traverse through there, including ones that go to, say, your pinky. So if I irritate or activate that nerve, you can sometimes feel it in your pinky. Right? You start to feel strange feeling in your pinky, and you also perceive as some of that's coming from the pinky, and that's because of the nerve that's traversing that area. Some of the sensory pathways. So we're going to talk about something called the dorsal column or the posterior column, and that's an area located in the spinal cord. So down here in the cartoon, you see we have just a reminder, here's our sort of butterfly-shaped gray matter, which contains the cell bodies. And it's surrounded, which you can see here in yellow, the sort of yellowish region around it, which is our white matter. All right, so remember, we're in the spinal cord, the white matter is in the periphery, and the gray matter is more central. There's your dorsal root, with the dorsal root ganglion, where the cell bodies of the sensory neurons are located. There's the ventral root. And so we talk about dorsal column, we're talking about the dorsal aspect of the spinal cord, which is right here. I'm going to circle that in red there. So that's that white matter, the white matter zone, that's going to have axons that are ascending to the brain, carrying sensory information. So we call that the dorsal column or the posterior column. Once it gets to the brain, it's going to take a different pathway, which we call the medial lemniscal tract. And so we'll talk about these pathways more in depth in a couple slides. And then the lateral spinothalamic tract, which is really the second type of tract. So we have the dorsal column, which I'm going to review with you, and then the lateral spinothalamic tract. These two types of tracts carry different sensory information, and it's important for us to understand these pathways because, clinically, when things like strokes or spinal cord injuries can disrupt certain pathways or different regions of the brain or spinal cord, and then we can have, because of that disruption of the sensation modalities that come along with it. And so we'll talk about that. And so this is from your notes. So for example, sensation going through the dorsal roots, entering the spinal cord and ascending to ultimately synapse in the region of the medulla, the brainstem. Then you see it crosses over and then ultimately goes to the thalamus and then to the cortex. And so we're going to talk about the different pathways that sensations take to get to the cortex to be perceived. Okay, so sensory information when it's reaching the cortex, right, it's primarily going to go into that post-central gyrus, which I referred to earlier. That post-central gyrus All right, you can see it highlighted in blue in the cartoon over here. Remember, it comes just posterior to the central sulcus, which separates the frontal cortex from the parietal cortex, or the parietal lobe. Here, highlighted in red is just really covering motor, so we're not going to be covering that in this lecture, but sensory is in blue. And so again, that's in that somatic sensory cortex, that post-central gyrus. Now, they're only showing you one hemisphere, all right? So like maybe the left hemisphere, all right? Where there is a right hemisphere here, they're just not showing you that. So in this hemisphere here, what you're seeing is this cartoon that's laid out on top of the cortex there called the homunculus. And the homunculus is really just a drawing to show you the region of the cortex that is responsible for interpreting the sensory information from that part of the body. So for example, sensations from the face, right, you can see that the homunculus designates this region right here. So this is sort of the real estate within the cortex there, the region of the cortex that will be interpreting and receiving information from that sensation from the face. You see the hand, you see the feet, and so on. You notice how like the lower extremities and the legs over here are more medial, so this is actually more central. This portion of the cortex is more lateral, so the feet more central, and we move across the cortex over here, so it's more lateral. So like the tongue and the face will be over in this region. And so, so it has this particular design to it, so where the lower extremities are more medial, then the body, you can see sort of the head, the hands, and then the face, and so on. And so what I really want you to kind of get from this is that these homunculus, you'll notice the hand and the face are particularly large, or large regions of the cortex that are designated for sensory information from those regions. Because we have a lot of sensory neurons, a high density of them in those regions to help us discriminate various types of sensory modalities. Right? We have to have a lot of sensory modalities within our hands and within our face. And so there's a large real estate within the brain that's designated for that. Whereas, say, in our trunk, we need really not as much discrimination in terms of different types of touch and tactile sensations. And so therefore, that's a smaller region of the cortex. So coming back to the pathways, all right, that dorsal column I said was the first one. So the dorsal column pathway, that's located in what we call the posterior funiculus. A funiculus is really just a term to designate the entire white matter section of the spinal cord there, which is located posteriorly. I've highlighted it in blue for you. And we can actually further designate it. I could draw a little line down the middle of it. And so this would be, the posterior column for one side of the body, and this would be the posterior column for the other side of the body. Okay, so for example, if I have sensory information coming into the dorsal root here, and it goes up the posterior column, it's going to go up on the posterior column on the same side of the body. Okay, so the right side of the body has the right columns, and the left side has the left columns. They're separated from each other. Now, in terms of that sensory information that goes into those that posterior column, all right, they're specific. We have what we call conscious sensations. So we have what we call light discriminative touch. So this is anything from just light touch, okay? But categorized within light touch, we have a couple other things, like 2-point discrimination. 2-point discrimination is being able to tell when two things are touching you at once. So if I had two pins, for example, and I was touching your finger with it, you'd be able to tell me that there's 2 points, 2 pins poking you in the finger. What I would do is I'd move the pins closer and closer together until you only felt one. So really with 2-point discrimination, the closer I get those pins together, the better your 2-point discrimination. Meaning you're still feeling 2 points as they get closer and closer and closer together until they're so close together, you only start to feel one at that point. All right. The reason we would have that is because in the fingertips we have very high density of touch neurons. So high density, which means every single neuron that's there carries its own path and tells the brain that it's coming from that particular neuron. So this is what enables me to feel 2 points, is that I'm stimulating 2 separate neurons. So having a very high density or cluster of them means that I can get very close together and still be stimulating separate neurons. However, when they get close enough together, then I'm really kind of stimulating the same neuron. If I stimulate the same neuron, I get the sensation of only having, only 1 point. Compare that to, say, the back, all right, or the chest or the back, where if I bring those points together, I might feel it when it's wide apart. I can feel 2 points, but there's not as high a density of the neuron. So when I actually start to bring them closer together, I can't get them as close because there's not as many neurons. So the fewer neurons that there are have to cover a larger area. And so what that means is that, when I come closer together, maybe they're still pretty far apart, I'm starting to sense that as one. You guys can test that out, on siblings or, significant others if you have them. And just, you check out this 2-point discrimination. See that the back, for example, has far fewer neurons. And so 2-point discrimination is not as good. Stereognosis. So stereognosis is basically, without looking, being able to feel different objects, the shape of them, the weight of them, the texture of them, and being able to figure out what those objects are. So in some cases, you might go to a doctor's office and you can put your hand in a box and feel around and say, what is that? If it's star-shaped or circle or square shape. Other cases, you can hand them keys or a pencil or a wallet, and they can discriminate what those objects are. Graphesthesia. Graphesthesia is like if you have the person's eyes are closed, you have their hand, and as the photograph is showing, if I draw or sketch something into their hands, I'm not actually, in this case, they're showing like a figure 8. You're not actually drawing it. You're just basically, outlining. You use it with your finger. Just outline the figure, figure 8 or any type of shape or letter. They can tell you what that shape or letter is by just feeling it. Then there's pressure, which is just like a deeper touch. Proprioception, which is our awareness of our body in space. All right. So, for example, if I tell the patient to close their eyes, and then I can take their toes, for example, and take their toe, and I can lift the toe up, or lift it down, and when I lift it up or down, I can ask them, is it up or down, and they'll tell me, right? So they're aware of where their body is in space. And then vibration, right? Using tuning forks and so on to feel different frequencies of vibrations. And so these are all the different modalities of sensations that are going to enter into the spinal cord and go into this dorsal column to ascend. Okay, so again, touch, proprioception, vibration will all be carried into this dorsal column. So I'm using these sort of block diagram here to show you how the neurons go from the signaling, from the sensory signal to the brain, to the cortex. So we start with the blue box down here. So cell number 1 is going to be the sensory neuron. We call it the first order neuron. This is the one that has the specialized receptors to pick up the different sensory modalities. And so in this case, we're talking about the posterior or dorsal column pathway. So that's going to be touch, vibration, proprioception, etc. And so that's going to enter into the dorsal root, right? Its cell body is going to be located in the dorsal root ganglion. And it's going to enter into the spinal cord. When it enters into the spinal cord, it's not going to synapse. Instead, it's going to go into that posterior column on the same side of the spinal cord. So if it entered into the right side of the spinal cord, it's going to go up that right posterior column. And it's going to ascend all the way into the brainstem in the medulla. So that's why I say here's cell number 1, first order neuron, gets into the spinal cord, stays ipsilateral, stays on the same side of the spinal cord, all the way to its synapses on cell number 2. So that's our first synapse. Cell number 2 is located in the medulla, in a particular nucleus called the posterior column nucleus. All right, so that's located in the medulla. So that's where this cell is going to synapse. So for example, if I have the sensory neuron is from my fingertip, that's going to go from my fingertip into the spinal cord all the way to the brainstem. So that's one cell. All right. Then in the medulla, I have cell number 2, and that's nucleus, and that's going to give off its projection or its axon, which is going to become part of what we call the medial lemniscus. And you'll notice I drew the arrow moving to the other side, so this is the contralateral. So cell number 2 sends its axon to the opposite side of the brain in the medulla, in the medial lemniscus. That's going to go to the thalamus on the opposite side of the body, in the brain, in what we call the VPL of the thalamus, the ventral posterolateral nucleus of the thalamus. This is cell number 3. Cell number 3 from the thalamus will then go to the cortex, and it will send that information to the cortex via what we call the internal capsule, which is really just a highway for the axons to go to the cortex from the thalamus. And in the cortex, it'll go to the region of the cortex for that particular part of the body, all right, which was designated by what we saw with the homunculus. So there are essentially, for any type of sensation, we're talking about touch, proprioception, or vibration, there are 3 neurons that are all going to carry that information all the way to the cortex to reach that final and fourth neuron. So if we're going to look at a cartoon of that to kind of give us a good visual of of the pathway I just talked about. Again, we're talking about the posterior column pathway. And so the sensations, again, that's our touch, vibration, proprioception, and so on. So you see it anywhere from the body. So you see the upper extremities, the torso, the lower extremities, even the face. Face would be carried by like cranial nerves, though, in this case. And here's the cell bodies in the dorsal root ganglion. So this is dorsal root ganglion cells, so cell number 1. All right, picks up that sensation, let's say, from the leg, carries it into the spinal cord, then up the spinal cord, all right, in the dorsal column, all the way into the medulla where it synapses for the first time, and it will synapse with cell number 2. So cell number 2 is in the dorsal column nuclei. Specifically, there's the nucleus cuneatus and gracilis, which we don't have to get into right now. All right, but these are the dorsal column nuclei. And from there, the cell number 2, you can see that it crosses in the medulla to the opposite side of the body, to the opposite side of the brain, and ascends to the thalamus. So that's the thalamus right there. So that's where cell number 3 is. So this pathway from the medulla to the thalamus, that's referred to as the medial limniscus. That's the medial limniscus. Cell number 3 is in the thalamus, and the thalamus is like a relay, and that's going to relay the sensory information to the appropriate parts of the cortex. So here you can see again the homunculus. So for example, I circled the leg down here. So in the thalamus, that would send that to sort of the medial region of the cortex there, in that post-central gyrus. And so that's where it can then be interpreted with neuron number 4. The next pathway I want to talk about is the spinothalamic pathway. The spinothalamic pathway carries information primarily for pain and temperature. All right, so pain and temperature. If we look at the cartoon here, the path that's going to take you to the spinal cord as it's ascending to the brain is not the dorsal column. All right, however, it is in what we call the anterolateral or spinothalamic pathway. So again, the funiculus is a sort of a major region of white matter in the spinal cord. There was the posterior funiculus back here. Highlighted in red over here is the lateral funiculus. And then over here is the anterior funiculus. So the combination of the anterior and lateral, so it's in the region that spans both. We call it the anterolateral pathway or the spinothalamic pathway. So we're essentially going from the spine to the thalamus here, but it's in the anterolateral region. So over here, which you can see highlighted for you in red, that is the spinothalamic pathway, where these pain and temperature neurons are going to ascend and go to the brain. Ultimately to the same destination, which is going to be the thalamus and then to the cortex as well. So we call it the anterolateral spinothalamic pathway. Over here, you can actually see the part of the dorsal column there. All right, it's divided into what we call the gracilis and the cuneatus, which again, we don't need to know for our purposes here, but we need to know that that's the dorsal column. That dorsal column will be carrying that information for touch, proprioception, and vibration. The anterolateral will be carrying pain and temperature, so they're separated from each other in the spinal cord. Why does it matter? Well, clinically, if you say you had a stroke that only affects the anterolateral part of the spinal cord, you could lose pain and temperature sensation. Or you had transection of your posterior spinal cord, you're going to lose, proprioception, light touch, and so on. So, you can have focal injuries and lose different types of sensation. And we can actually figure that out without doing any imaging at all, just on a test. By testing different sensory modalities, we can see if there's deficits. And that can lead to us kind of figuring out, even localizing where that damage might be. So the spinothalamic pathway, so similar to what I showed you with the dorsal column pathway, I'm just going to use this sort of box diagram here, where blue is going to be cell number 1. That's coming from our dorsal root ganglion. So again, that's the one that's picking up the painful stimulation. And so the biggest difference is going to be here is that the pain neurons, when they enter into the spinal cord, okay, so if let's say I have pain in my right hand, it's going to enter into the right side of my spinal cord. When it does so, it's going to synapse immediately. Unlike what we saw with the dorsal, where it goes all the way to the brainstem, that doesn't happen in this case. In the spinothalamic, it's going to synapse onto cell number 2 in the spinal cord, in the gray matter. So we call it the spinal cord nucleus, so the gray matter, that central region of the spinal cord. It's going to synapse there, and that cell is going to send its axons across the spinal cord, across a structure called the white commissure, which is in the anterior portion of the spinal cord. And that's going to then ascend all the way to the thalamus on the opposite side of the body, again in the VPL. And then cell number 3 in the thalamus is going to ascend all the way to the cortex for processing again in that postcentral gyrus. So to give us a visualization of that pathway, we have, again, we're carrying pain and temperature in this pathway. So cell body is located in the dorsal root ganglion, as it was for the other types of sensation. However, when it enters the spinal cord, it synapses right away. When it synapses there in the gray matter of the spinal cord, it's then going to cross the spinal cord at the same level it came in at, and then that's going to ascend, and we call that the spinothalamic pathway in that anterolateral region of the spinal cord. And it's going to ascend all the way to the thalamus on the opposite side, where there is cell number 3, again in the VPL of the thalamus. And then that's going to send that to the appropriate portion of the cortex there, as designated by the homunculus, which you see there. If we're to look at the gray matter in the spinal cord, what you're seeing here is, for example, here's a dorsal root. Here it's entering into the spinal cord, and so this whole gray area, all right, represents that sort of butterfly or H shape. That's the gray matter in the spinal cord. And so when it enters in there, there's cells that are located in designated regions in that, what we call that dorsal horn of the gray matter there. So it can be classified as certain regions, like the substantia gelatinosa or the nucleus proprius, which just basically codes certain regions of that area, or in layers, which are also designated by Roman numerals. We're not going to get into the details of that, but ultimately it's just to say that there are different regions of that horn there that are going to interact with different incoming neurons, different sensory neurons. So in this case, it's this particular pain neuron is coming into the spinal cord, interacting with the dorsal horn and the substantia gelatinosa, where cell number 2 is located. And that's going to send its axons across the anterior aspect of the spinal cord, right, across what we call that white commissure. And then it's going to go into the anterolateral path over here, the spinothalamic tract, and it's going to ascend to the thalamus on the opposite side of the brain there. So those are really the 2 general pathways that I want you to know. The spinothalamic pathway, carrying pain and temperature, and the dorsal column medial lemniscus pathway, which carries light touch, proprioception, vibration sensations. Okay, now what I want to do now is get into a little bit more detail about pain itself. Okay, so with pain, again, there's differences in the type of pain. So I mentioned before we have nociceptors, which are really just the specialized pain receptors that really pick up on mechanical type pain, chemical type pain, or even temperature related pain. They don't have any kind of specialized nerve endings that you might see in different pressure type receptors, where there's like Merkel discs or Pacinian corpuscles. Instead, they don't have any of those specialized bulbous endings, so they just call them free nerve endings. And a really important designation for pain is fast pain and slow pain. So fast pain, neurons that, neurons are designated by really the diameter of their axons and the amount of myelin that they have. So we'll usually say if there's an A-type fiber, that means it has a very large diameter and lots of myelin, which means it has a large or very fast conducting velocity. So we refer to those as A-type fibers. And then B-type fibers would be sort of intermediate, and then C fibers would be slow because they're usually smaller diameter, and no myelin. So this C would be like the slowest type. And then there's sub-designations within each, which we're not going to get into a lot of detail on. But for example, you have A-alpha fibers, A-beta, A-deltas, and so on. And that's just sort of, sub-designations within them. And so, for example, fast pain and slow pain are A fibers, specifically A-delta type fibers. And so you can see that over here. Here's A-delta. So that's a fast type. What do we mean by fast? Well, fast means it's conducting very quickly, right? But what type of pain is transmitted that way is sort of a sharp, pricking, acute or electric type pain, stabbing type pain. And importantly, it's usually very well localized. So if you have stabbing pain, you can usually point to the part of the body where it feels like you're having stabbing pain. Like you can be very specific about it. So localization is very important when it comes to differentiating types of pain. So fast, again, sharp stabbing type pain, easily localized. Now that's helpful for us because that can be very damaging to the body. And so being able to sort of pick up on those signals very quickly, we can run reflexes with that and move our body and get it out of harm's way. And it's going to send those really quick signals. Slow pain, on the other hand, slow pain are the C fibers. So these are the smaller ones that are unmyelinated. This is like the slow, aching, burning pain, that throbbing pain, something that it's associated with the pain of nausea and so on, and often associated with patients who have chronic type pain. That sort of unbearable type pain. That's often associated with that slow pain. Importantly, with slow pain is diffuse and not well localized. Right? For example, like when people feel they have belly aches or stomach aches, right? They're usually like using their whole hand to kind of designate an area, like, oh, my area over here, or right around my belly button, or my chest, I have, having heart pain or anginal pain, usually doing this type of thing. Where that's not really localized, it's more diffuse. Whereas stabbing pain, they can point to it, and it hurts right there, right? So this is the difference in fast versus slow, okay? And in point of fact, we have both when we get injured. So for example, if I step on a tap, that would be very sharp, pricking type pain, right? I'd have a reflex to withdraw from that, but let's say it's stuck in my foot, I pull it out, I initially get that sharp pain that hurts, that makes me react quickly, and that happens within seconds, and then a few seconds later, or even a few minutes later, you start to get that sort of slow, aching, painful feeling that surrounds that area. It's not well localized to where the puncture occurred, but might be a much larger circumference or a much larger area around your foot that starts to feel achy and throb. And so that's the slow C fiber type pain. Okay. And again, the receptors that pull it off are mechanical, thermal, or chemical in nature. When it comes to fast pain is either mechanical or thermal. All right, so extremes of temperature or extremes of pressure and so on. Slow pain can be all three types. It can be mechanical, thermal, or chemical type pain. When we talk about chemical type pain, there's certain mediators that can initiate pain, like pain from inflammation, for example, like bradykinin, histamine. You can have excess potassium can do it, proteolytic enzymes. These are all things that can actually trigger this chemical pain, these slow fibers. As you see in the cartoon over here, the A delta, which are the fast, well localized mechanical or thermal type stimulant, and then there's the C fiber, which is going to be that slow, chronic. They connect the chronic kind of pain. That's you'll notice that when they go into the spinal cord, they both synapse right away, as we talked about. They're synapsing in different layers there, as designated by the Roman numerals, which you guys don't need to know right now. But they synapse in different layers, and then they cross over. As I said, they cross the spinal cord and go up the opposite side of the spinothalamic path up towards the brain on the opposite side. Okay. So the last thing I want to kind of mention before I move on is that with fast and slow pain, in the surface of our body, like in the skin, we have both like fast and slow type pain, as I mentioned with the example with the tack. On the other hand, when we start talking about the viscera or the organs in our thoracic or abdominal cavity, they really are not well innervated with fast pain at all. So we don't get really well localized pain from the organs. Instead, we get more of the slow pain fibers. Okay, so it's more diffuse. So we're going to get more into that, where the viscera carry the primarily slow pain, and the surface of our body carries both. So I want to talk about sensitization and adaptation. So when it comes to pain, the neurons can sensitize. And what that means is that for a given stimulus, a painful stimulus, the next time that stimulus comes along, it's more pronounced, it's more sensitized to that. So in fact, the term for that is hyperalgesia. So for the same given noxious stimuli, you're getting a more pronounced effect. And the reason for that is because you have what we call sensitizing agents. Sensitizing agents like prostaglandins and substance P. So for example, you could stimulate the pain neuron, and it may release glutamate as its neurotransmitter, and then that sends the signal up to the brain and so on. Glutamate is one of the primary excitatory neurotransmitters. Now, in addition, when it's releasing glutamate, it can release substance P. Now, substance P, when it's released, its duty there is not to act as a neurotransmitter might, but instead acts locally on the presynaptic and postsynaptic neurons and causes rearrangement of receptors and so on. And this can cause sensitization of those so that the same signal causes a more pronounced effect if it comes along again. Now, in cases of that, where we'd see that would be oftentimes with these sort of slow C fibers. So, for example, allodynia. Allodynia is the next one on the bullet list there. Allodynia is where you have a non-noxious stimuli causes pain. So, for example, in our gentleman here, who's our Batman, right? He's got this sunburn there. If I touch the skin of somebody who's not sunburned, right, that doesn't cause any painful stimulus. It's just light touch. On the other hand, somebody who's severely burned like that, if you touch the skin lightly, they might jump off the seat, right? They might scream in pain. And so this is in part due to this increase in sensitization. We call this allodynia, where this isn't a painful, this isn't a noxious stimuli, but it's causing pain because of the burn. The neuropathic pain. Neuropathic pain is really something wrong with the neurons themselves, where they may be firing inappropriately, causing pain. So this is where there is no noxious stimuli at all, and you're still having pain. Now, some of the chemicals, so I mentioned, there's chemoreceptors that can cause pain. So we said temperature can cause pain, extremes of mechanoreceptors like deep pressure can cause pain. Chemicals like bradykinin, histamine, proteolytic enzymes can also induce pain. And they've done some very interesting experiments where they've given some of these chemicals subcutaneously, and it has induced serious pain. So they see that these chemicals themselves can elicit pain in neurons when there wasn't any other real noxious stimuli other than the presence of these chemicals. Now, finally, adaptation. When it comes to adaptation, this is really talking about the neuron's ability to stop firing when the stimulus is present. So if there's a stimuli that's there, right, the neuron will start to fire action potentials. And if the stimuli remains there, ultimately the neuron adapts so that the action potentials fire less and less frequently, and ultimately until it stops. And this is useful for certain sensations, like certain touch sensations on our body. Like if we're wearing our shirt, we put our shirt on, we're aware of our shirt, but then throughout the day, you're not really aware of your shirt touching your skin because it's sort of adapted and reduced its number of action potential firing. But when it comes to pain, pain doesn't adapt in that way. Instead, it continues to fire, and in cases like this where it's sensitized, it can even amplify its firing. And the theory behind that, or the reason behind that, is that we don't want our body to, or we don't want our brains to ignore painful stimuli because we want to protect our body. So we don't want that to reduce its action potential firing and then we can ignore it. Instead, we want to make sure we change our positions or remove the substance that's causing the pain, so we're aware of it. Okay. So, for example, as you're sitting here listening to hours of my talking endlessly, the tissue overlying your ischium might actually start to be irritated. That's because you're compressing it, causing some ischemia in that region, which is activating these pain fibers. And so, to adjust that, right, you just shift your body weight around, you move around a little bit in your seat to alleviate some of that and reduce some of the pain. But if you just stay there, the pain continues and you start to notice it more and more. It doesn't just go away. And that's to make sure we're aware to move our body. So now fast and slow pain. All right, when it comes to fast, that's essentially going to follow the pathways that I've already talked about. It's that whole spinothalamic pathway to the thalamus and then to the cortex. And the primary neurotransmitters for things like fast and slow are going to be glutamate, and then the sensitizing agent being like substance P. With slow pain, however, those C fibers, with slow pain is a little bit different. So slow pain will synapse in the spinal cord at the same level it enters, It'll cross over in that anterior white commissure and then enter the spinothalamic pathway and ascend. And instead of going to the thalamus, where it differs is, it's actually going to branch off and go into and synapse with a lot of different neurons in what we call the reticular formation. Now, the reticular formation is important for our state of arousal, consciousness, our ability to attend or pay attention to things. We rely on this reticular formation. So the reticular formation is really good at sort of filtering out noise and helping us sort of pay attention to certain specific things in our environment. So this actually, the slow pain fibers branch diffusely throughout the brainstem, particularly in that reticular formation. In fact, most of the fibers will synapse there as opposed to the thalamus, maybe 90% or so. 10% will go to the thalamus and to the cortex following the typical pathway. But the rest of them are diffused throughout the brainstem regions here and these lower areas of the brain. And this is an older area of the brain. And so most of it doesn't reach the cortex. Okay, so in fact, if you were to disconnect the cortex from the brainstem, you can still have this slow chronic type pain, this very severe type pain. Okay. So again, that reticular activating system, it's important for us to attend to certain stimuli while filtering out some others, because we're bombarded by, hundreds of different stimuli all the time. So we need to be able to kind of tease apart what's important and what we need to pay attention to. And we're getting stimuli from, visual centers, which you can see over here, visual centers, auditory, and so it's taking in all these sensations, going into this greenish area, which is the reticular activating system, and it kind of has to filter out some of that, quote unquote noise, if you will, and then send, signals to different parts of the brain that we have to really attend to. So when it comes from, touch or pain receptors from those slow type fibers, this is important because this is actually exciting the reticular activating system so that we attend to or pay attention to or are conscious of the pain. So this, again, that sort of diffuse, slow type pain, we can't really ignore it because it's interfering with our activating system. It's making us pay attention to it. This is also why it's difficult to, say, fall asleep while you're in pain, right? Because it's trying to make sure that we're attending to it or paying attention to it. Now, pain is clinically difficult to assess in terms of the level of pain because it's very subjective. People have different pain tolerances and perceptions of pain, and part of that is due to the fact that we actually have built-in analgesia. We have built-in ways of reducing some of our pain. So in other words, we don't want our pain levels to go so high where we can't really focus on anything else that would help us survive. So we're going to have some feedback that kind of helps to suppress some of that pain. Not completely get rid of the pain, but at least subdue it, okay? So that we can continue to kind of function. And we have these built-in analgesia systems. So if you look over here, just so we're aware, I just want to make sure we understand what we're looking at. This is the midbrain. All right, here's the pons, and this is the medulla. So this whole area right there, that is the part of the brain stem. And so superior to that would be like the cortex up there and the thalamus and so on. This little blue region, all right, is called the periaqueductal gray. And so these cells that are located, these neurons that are located there, will actually send axons to a region in the pons known as the nucleus raphe magnus. And its neurotransmitters known as an enkephalin. So these enkephalins are released onto this neurons located in the nucleus raphe. Then these cells will then send their axons to the spinal cord. So these will descend into the spinal cord and synapse on these little red interneurons. They release serotonin onto these little red interneurons, and these little red interneurons will then release enkephalins onto both the presynaptic and postsynaptic pain neurons, both fast and slow. And so these enkephalins are going to interact with receptors located in the presynaptic, postsynaptic sides of these pain neurons. And enkephalins are a type of what is known as endorphins. So endorphins there's a whole category of these types of substances that interact with what we call opioid receptors on these neurons. Opioid receptors, when activated, suppress painful signaling. Just like we think of opioids or morphine, for example, like morphine can reduce pain, right? So it acts on these opioid receptors. We have endogenous morphine in our body. In fact, that's what the term endorphin comes from. Endogenous, meaning we make it, and the orphans, so the endorphins. So this is our endogenous morphine. So enkephalins is one of these types of endorphins, and it suppresses pain signaling. Okay, so it suppresses that pain signaling, so that helps to reduce the amount of pain we have, and that can help kind of modulate that level of pain. In fact, one of the hypothetical examples of this is when some a runner gets the runner's high, like running a marathon. What they found is that in some cases their endorphin levels may be very high, which is like high levels of their endogenous morphine, and that those levels may rise to help modulate the pain from all the inflammation and from the running, and so this may help to suppress some of that pain. In addition to having that sort of descending pathway of pain suppression coming from that like periaqueductal gray region, we can also do what we call tactile sensory signals. All right, this is called the gate theory. So in gate theory, what this means is, like, for example, if I were to bump my head or my elbow on something, one of our first instincts is to then rub that region, right? So if I bump my head, I want to rub my head or rub my elbow or rub the area that got hurt. And in fact, there's a reason for that we do that. It's because rubbing something makes it feel better. And what they found is that when it comes to, say, that slow type pain, so you bump your head, you get that sharp pain from hitting it on the edge of something, but then what follows later is a little bit of throbbing and achiness, and if I rub it, it feels a little bit better. So we can inhibit that slow type pain by rubbing it, because what they found is that in shared nerves, so remember here's our dorsal root ganglion, those dorsal roots have mixed types of sensations going through there. So tactile sensations as well as pain sensations and so on. And so if we're rubbing it, we're really activating those touch sensations, which are those like A-type fibers, are very fast. And when they enter into the spinal cord, in this case it's branched. One part of it's going up, another part of it is actually going to connect with the synapse coming from this slow fiber there. So it's going to modulate the pain fiber, so that's slow pain fiber. So when we're rubbing it there, we're sending very quick signals via touch into the spinal cord, and that's giving a branch to the slow pain neuron, and it's going to inhibit its firing. So there's actually an inhibitory signal that's there. So when we rub it, we're actually inhibiting some of the slow pain fibers. And so that's why it helps. In addition, we can use something called transcutaneous electrical nerve stimulation, which you see over here. In short, called TENS, and that's really putting like an electrical stimulating pad over a certain area, and by using the stimulation from that, we can activate those sensory neurons, and it creates an analgesic type effect by reducing pain in certain areas, and it can be helpful for certain types of pain. In other cases where you may have more intractable pain from, say, cancers or something like that, you may have to resort to things like cordotomy, where you actually go in and sever parts of the spinal cord where there's the pain pathways, so that you can stop the pain, or even using ablation. In some cases, using electrical stimulation of various parts of the brain. For example, you could actually stimulate the periaqueductal gray to create that increase in endorphin release to help subdue some of the pain signaling as well. So there have been multiple ways of trying to alleviate pain. All right, so referred pain. We're talking about referred pain. So you guys probably know what this is, but we're going to talk about it more specifically. For example, when it comes to, say, a heart attack. When somebody has a heart attack, they may clutch their chest, right? And again, they're not pointing at a specific region, but there's this diffuse, slow type pain, which is causing this sort of crushing substernal pain that they get. They'll make a fist like that, right? But at the same time, they may also be feeling their left shoulder, their arm, and even their jaw can start to feel pain. And in fact, the only true pain is coming from the heart, but they're feeling it in their jaw and their arm, and that's called referred pain. So this is where an organ in the body that's having pain is it's causing referred pain to the surface of the body. So we're feeling it on the surface of the body, even though there's no damage to the surface of the body. Okay. So again, that's called referred pain. And so part of the reason for this is developmentally, the organs, when they're developing, they're going to share. So over here you can see the part of the body, skin, nerve fibers entering a certain segment of the spinal cord, and the visceral nerve fibers. So they'll share a certain part of the spinal cord when they enter, and you'll see that there is crosstalk between them. So say we have pain fibers coming from the surface of our body, they're going to interact with the same ones that the viscera are going to connect with. And so since they're sharing some of the same neurons, it's being perceived as coming from the same areas of the body, either from the skin or from the organ. So for example, again, if the heart is in pain, we're going to also perceive that it's coming from the surface of our body because it's sharing the same neurons there. And so this is why we have this referred pain. And the same thing happens again. The heart's sort of the classic example, but what you're seeing in the cartoon over here is various other parts. You see broad regions like the stomach, for example, a large region there, which is causing more of a diffuse pain that we feel on maybe even the surface of our body. Right around the umbilicus there, and you see the ureter and so on. So these are all just regions that help, that cause the sort of referred pain. So again, it's really the organ that's been damaged or that's signaling the pain fibers, but we're referring it to the surface of our body because of this shared pathway. Now, when it comes to visceral pain, I mentioned this before, it doesn't carry many fast fibers at all, so we can't really localize pain when it comes to the organs. It's more diffuse than that. So they're all carried essentially by those C fibers, those slow type pain fibers. So localized damage to an organ doesn't actually cause severe pain. So for example, if you're doing surgery on somebody and they happen to be awake and you're working on their intestines, if you accidentally slip with a scalpel and puncture their intestinal wall, they're not even really going to react to it. All right, you can stab the intestines there. They really don't react to it because there's not much in terms of those sharp, fast type fibers at all. So they're not going to feel it. Okay, and so in that case, you're not going to get a reaction out of them. On the other hand, if you activate the C fibers, this is more of a diffuse stimulation of pain fibers. So things that come from ischemia, from lack of blood flow to a region of the GI, chemical damage to the surface like protons, like in causing ulcers in the stomach, spasms of the muscles that surround some of these hollow organs, distension, any kind of stretching of the connective tissues that are heavily innervated by these slow fibers. This can cause severe pain, like excruciating pain, but it's usually more like diffuse, has to be a more diffuse stimulation, and it's not well localized again. So I can't point with my finger and say this part of the duodenum is hurting. It's really like you're kind of using your hand saying, well, this area is hurting. So that brings me to what we call parietal pain versus visceral pain and helping us localize pain in the patient. When we talk about parietal pain, what we're really talking about is pain from the surface of the body, including the parietal pleura, all right, or the parietal peritoneum, parietal pericardium. So remember those connective tissue layers that surround the organs? The parietal layer of those have both fast and slow type pain. So from the surface of our body to the parietal layer that's around the organs, all have fast and slow type pain, which means we can localize pretty well with the fast, all the way to the parietal layers of the pleura, pericardium, peritoneum, and so on. So we can actually localize it. So for example, if you have pain in your heart or your lungs, it's going to be more diffuse. There's no fast pain fibers there. That's just visceral pain. It's more diffuse. So if they have, say, pain in the lungs or the heart, they're going to be giving you more of a diffuse picture. But if whatever's causing that pain has extended all the way to, say, the parietal surface, right, the parietal pleura, for example, in the lungs, now you can actually have parietal pain. This is going to have fast fibers as well, just like the surface of the body, and so they can have, that parietal pain. So they can actually localize it then. We call that like pleural pain. So somebody's breathing in, they may have like pneumonia and they didn't have any real pain at all, and then that extended to the parietal surface of the pleura, and now they're having pleuritic pain. So every time they breathe in, it's sharp, and they can actually point to it because it carries those fast fibers. But the lung parenchyma does not. Okay, the heart does not, so we can't be that localized with it. So that's why I have said here, parietal surfaces like the skin are supplied with extensive innervation from the peripheral spinal nerves. Meaning, so it follows that dermatome. So we've talked about the dermatomes in previous lectures. So surface of the body. Anytime we irritate the skin or the parietal surfaces of those pleura, that's going to go into the spinal cord segment that's associated with this specific dermatome, right? Now, that's going to help us kind of localize. Now, when it comes to the visceral pain, again, that's again carried mostly by those C fibers, and we're going to have referred pain. So when the organs get injured, they're going to send pain information back to the spinal cord via the autonomic nerve pathways. Remember that the sympathetic nerves, when they innervate the organs, they're going to go through the sympathetic chain, for example. So any afferents coming from, or any sensory modalities coming from those organs are going to go back and follow the pathway, like the splanchnic nerves, follow them back into that sympathetic chain, into the spinal cord segments designated by the autonomic nerves, T1 through L3. Okay. So I'd like to give an example of this. Let's say here in the cartoon we have somebody who has appendicitis. So here's the appendix down there, part of the colon, located in the right lower quadrant. And if it's true organ pain, so remember this is the appendix is part of the colon, which is the organ. If it's true pain there, so we're having ischemia of some sort, right? It's causing aching pain. All right, that's those afferents, the nerve fibers for that pain from the organ, is going to follow the trajectory back with the autonomic nerves, which is part of the chain over here. So you can see the sympathetic chain. And in this case, the appendix would go back and into the spinal cord segment of T10. So that's going to go back to T10, and any of the body surface area that also goes back in T10, any of our dermatomal that goes back to T10 is also going to go in at that segment, and we're going to get referred pain. So in other words, when the appendix is inflamed and actually causing pain, when it goes back to T10, the surface of our body that's innervated by the dermatomal distribution of T10, which is right around the umbilical region or the belly button region, that's going to feel pain. So you're going to start to feel this sort of diffuse achy pain right around your belly button, even though the appendix is in the right lower quadrant. And this is because of the referred pain of where this signal is going to in the spinal cord, which is right around the T10. So again, look at that dermatomal map. You'll see where T10 is. It goes right across the belly button. And so you're getting referred pain. So real visceral pain is going to be referred in some way. And so it's going to be right around the belly button. So that's the true visceral pain. Now, the parietal portion of this, you can also have parietal pain, so it's body surface pain that's real. And that may be coming from the appendix, the inflamed appendix, irritating the parietal peritoneum. So remember I told you the parietal portion of these connective tissue layers have fast pain as well as slow pain. So when it irritates the parietal peritoneum, that's going to irritate the body surface area that's designated by that particular dermatome. So you see the parietal pain over here. That goes back into L1. So the body surface that is innervated by that right lower quadrant region. So in that case, when you're feeling parietal pain or sharp pain in your right lower quadrant, that's actually coming from irritation of the parietal peritoneum. So this is where you have the achiness pain that's being referred to your belly button region. That's the true visceral pain. That's the organ itself that's suffering. And then you get sharp pain in your right lower quadrant. That's actually coming from the irritation of the parietal peritoneum by the organ. So you're getting pain in two different areas. And that helps us to kind of localize it to that region. So somebody coming in with right lower quadrant pain with also pain around the umbilicus, these are signs of appendicitis. So lastly, I just want to kind of touch on headache. Headache is really its own major category of pain and so on. But just to kind of give us an idea of what's causing the pain within the head. So for headaches, right, most of this is going to be referred pain. So something that's happening or causing pain inside the cranium, we often get referred pain to the surface of our head. And so things inside the cranium that cause pain are things like connective tissues like the meninges and blood vessels, like the middle meningeal artery is particularly sensitive. So if you get spasm of blood vessels, that can cause pain. If you get stretching or irritation of the meninges or the connective tissues in the brain, that can cause pain, which we then see that as causing headaches. And depending on what kind of tissue inside the cranium is actually being irritated, it'll refer to different parts of the surface of our head. So for example, down here color-coded, you see the falx cerebri. So this is like this, broad connective tissue band that actually sits in that longitudinal fissure that separates the hemispheres of our brain, the left and right hemispheres. And so that's densely innervated. And so when irritated, that can cause headaches. In green is the horizontal. It kind of spans, it separates the superior cranial vault, where the cerebral cortex is, from the inferior cranial vault, which houses things like the cerebellum. And so we call this the tentorium cerebelli. So that's more horizontal, separating again upper from lower. And if that gets irritated, that can cause headaches. And of course, any of the blood vessels that traverse there, which you can see some of the venous sinuses over here, as well as any arteries that are involved, can cause pain. And so for example, if I irritate the falx cerebri or anything in the superior vault above the tentorium cerebelli, the cranial nerve 5 carries pain innervation for that. And we refer that to the anterior portions of our head, which you see highlighted here in the cartoon there in blue. If it's below the tentorium cerebelli, which is the green thing, if it's below that in the inferior cranial vault there, that can be referred to the green area, which you see over here. So things like the brainstem, cerebellar vault, that can be referred to the back of the head. Any kind of eye pain or sinus congestions and stuff that can irritate the sinuses, that can cause pain in the face, right around the eyes, behind the eyes, and so on. So again, it's kind of important to understand the structures that have pain, because the brain itself, the parenchyma of the brain, does not carry pain fibers, or very, very little of it does. Okay? So intracranially, we talk about the connective tissues like the meninges. So meningitis, inflammation of the meninges, can cause severe pain, sharp pain, and diffuse pain. Low CSF, so cerebral spinal fluid. If you do, say, spinal tap to draw out cerebral spinal fluid, the cerebral spinal fluid helps keep the brain somewhat buoyant. And by removing the fluid, it loses some of that buoyancy and can actually pull on some of the connective tissue structures, and that can cause headaches. Migraine is sort of a topic unto itself. There are many different types of migraines, but one of the underlying causes is thought to be spasms of blood vessels. Spasms of the blood vessels can cause pain. Spasm of blood vessels within the brain can also cause some of those prodromal symptoms like visual changes, some of the sensations that people get prior to the headache onset. Alcohol, classic alcohol intoxication with headaches in the hangover phase. Alcohol itself, ethanol, can actually irritate the connective tissue in the brain and cause headaches. So it can cause it directly, as well as indirectly through causing dehydration. In terms of things outside of the cranial vault, so more on the surface, outside of the skull, we have muscle spasms. So people who are stressed out, in one position, they rub their necks a lot. That can cause headaches, like tension type headaches, temporal type headaches. Irritation of the nasal structures, I talked about that with nasal congestion. And then eye disorders, so any kind of visual disturbances can cause headaches as well. So I'm going to end with the pain lecture with that, and I will talk to you guys in the live Zoom session. See you then.